Hello, and welcome to the latest A Spotlight On interview. Today, we're going to be talking about single-cell proteomics. I'm joined today by Nikolai Slavov. Nikolai, could you please introduce yourself and tell everyone a little bit about what you do?
Thank you for inviting me. I'm delighted to join you. I'm an Allen Distinguished Investigator and professor at Northeastern University. My laboratory is generally interested in proteome biology, understanding how proteins, their interactions and modifications give rise to life, to normal physiology and pathology alike. And today we are going to discuss in more depth one particular research aspect, which is the technologies to quantify proteins in single mammalian cells that we develop and apply to various biomedical projects.
Thank you so much. And what drew you to focus on single-cell proteomics?
It is well appreciated and understood that most biological functions are performed by proteins, but our inability to analyze proteins in depth at high resolution has resulted in biomedical research being focused primarily on analyzing DNA and RNA molecules. And these are very fruitful research avenues. That is actually my background. I was previously focused on transcriptomics. I did my PhD in systems biology in David Botstein's group, where we used the technology of the day, DNA microarrays. But as we were studying that, oftentimes my data would suggest that what's the interesting biological processes that are taking place are happening at the level of protein synthesis or protein degradation. And we were limited in our ability to analyze those. And around 2011, I realized that existing mass spectrometry technologies can be applied fruitfully to analyze proteins with much higher throughput, quantitative accuracy and sensitivity than what has been done. And this particular opportunity wasn't being realized by anybody, as far as I could tell. And this made me think that if I'm able to realize it, I can help accelerate the development of this part of biomedical sciences. And I saw this as a very big opportunity. It resonated with me and with one of the most important criterions that they have for anything that they do, which is the ability to accelerate the development of scientific research, not only be one of the competent scientists who can help do a particular milestone research project, which is obvious to many other colleagues in the field, but can he do something unique that is going to make science proceed faster than it would have done without me doing that? And single-cell proteomics seem to be very much in this category. And I decided to give it a try to see if we can actually develop those technologies. The importance was very obvious to me and to anybody else that if we are able to achieve deep quantitative protein analysis of individual mammalian cells, that will have very many applications. So the significance was never in doubt. What at the time was much more in doubt and much more controversial is the feasibility to do that. Many of the leading experts in the field at the time believed that this is not possible. They were very skeptical. And I was a newcomer without background in that particular technology. So not surprisingly, I did not convince the leaders in mass spectrometry overnight that we can do something that they believed impossible. But I thought worthwhile giving it a try.
Absolutely. And you have developed these methods for high throughput single cell proteomics by mass spectrometry, your Scope 2 project, and you're using them to quantify proteome heterogeneity during cell differentiation. It's an emerging field. There's been a variety of methods proposed by different groups. So what makes Scope 2 kind of stand out from the crowd?
The approach of Scope 2 and other methods that we have tried to develop, something that we emphasize is its accessibility from the very beginning. I would also maybe take a step back just to give perspective on methods and how they have been developed and their differences. Because looking from outside, it is easy to see multiple different names being used and different methods. And the field appears to be quite crowded, but in a way, the methods that are being used fall into only a couple of categories, and the methods within a category are quite similar to each other. So one approach is to analyze one cell at a time. And in the jargon of mass spectrometry, we call this label-free because cells are not labeled. We analyze only one cell at a time. And this approach has been attractive to a number of colleagues. From my perspective, a major limitation of this type of approaches has been their limited throughput, because mass spec instruments are quite expensive and mass spec time is expensive. So if we only analyze one cell per hour, that is going to have limited scope of biological applications. And our strategy has been to use labels, which is oftentimes called multiplex approaches, so that instead of analyzing one cell, we can barcode proteins from different single cells with single cell specific barcode, and then we can analyze a dozen or more single cells at the same time. And at the end of the experiment, we can tell which protein came from which single cell and therefore do the single cell quantification. So this is one specific aspect that we introduced using isobaric mass tags. And until recently, all of the multiplex methods that anybody has used use the technology that we introduced at the time of using isobaric mass tags. More recently, we have introduced a different kind of multiplexing, which actually will be published in Nature Biotechnology tomorrow, which uses non-isobaric mass tags, and this has different set of advantages that we can discuss later on. But one aspect of our first approach of Scope MS and its second generation Scope 2 is the use of multiplexing, which allows to increase throughput. Another aspect at the time that was absolutely crucial is the use of an isobaric carrier, which is the fact that in addition to the single cells that we barcoded a small bulk sample of cells, which allowed us to reduce losses of single cells in various surfaces that the labeled set interacts with, and allowed us to increase our ability to assign amino acid sequences to the peptides. And that was key for being able to quantify proteins in single cells in the early days, in our very first attempt. At this point, new technology developed by my laboratory and other laboratories has made the use of isobaric carrier less essential, but at the time, this was the first demonstration, to my knowledge, of being able to quantify hundreds of proteins across hundreds of single cells. And that was very much enabled by using this approach of the isobaric carrier. And in fact, was enabled by using old equipment. We did not have access to cutting-edge, state-of-the-art mass spectrometry equipment. In fact, I did not have any equipment, so I had to collaborate with the facility at Harvard, with Bogdan Budnik, a good friend of mine at the time, who made a key contribution by providing access to equipment that I did not have at Northeastern. And the whole approach with the use of multiplexing, the isobaric carrier, and other aspects that we introduced made it very easy to implement in other laboratories. And that has been a guiding philosophy for us, not only develop the best methods that we can possibly develop to give us the most accurate, the highest throughput methods. All of that is great, but we have a significant constraint on everything we do. And that constraint is what we do should be reproducible in other laboratories. Others should be able to do it. And ideally, we make it as easy as possible for others by using equipment that is widely available, that is relatively inexpensive. They're not cheap. Nothing is very cheap. But at least we can use equipment that is much cheaper, in some cases orders of magnitude cheaper than alternative equipment. So that has been a very important guiding principle for us. Let's try to make this field, the technology, accessible. Let's try to make it as inexpensive as possible. Let's try to make it high throughput. And I think that these are distinctive aspects that have allowed many laboratories to adopt and start using Scope 2. And I know now of a number of mass spectrometry facilities, both in the US and in Europe, that have successfully implemented Scope 2. There are a number of other methods using multiplexing, sometimes with slightly different names, but they're really variations on the same approach that Scope MS introduced. I mentioned other methods that exist. There are label-free approaches, and another set of approaches that we recently introduced that I mentioned is by doing data-independent acquisition combined with multiplexing, non-isobaric labels used in that context. And I'm very enthusiastic about the potential of these approaches to inherit many of the advantages of Scope 2 in terms of being accessible, being relatively inexpensive, but they have the potential to provide even deeper and substantially higher deeper proteome coverage and substantially higher throughput than what the current generation of methods has achieved.
Thank you so much. And you've talked about the benefits and potentials there. What about some of the challenges? What are some of the key challenges in scaling up single-cell analysis to the proteome?
Some of the challenges are very similar to the challenges of mass spectrometry proteomics in general, not only single-cell analysis, but any kind of mass spectrometry proteomic analysis is not as widely integrated with biomedical research as DNA, RNA sequencing methods. And the reasons for that are numerous. Some of them are technological. I think a lot of them are societal and policy-based. It's a level of understanding of the technology by colleagues who drive biomedical research. It's funding from various government institutions and so on. So all of these problems that have generally made mass spectrometry proteomics less accessible and less integrated with biomedical research are also applied to single-cell protein analysis, unfortunately. These problems are not insolvable. They certainly have solutions. They're not simple. I cannot solve them overnight, but we try to help with those. Certainly at the level of education, we are very passionate about doing our best to explain the technology in accessible manner to the wider community. Some of those have to do with articulating compelling vision and justifying funding to develop standard operating procedures and articulating which are the problems in biology and medicine that really need proteomics and why we should invest in doing the protein analysis as opposed to focusing on the more accessible transcriptomic genomic analysis. In terms of adoption of Scope 2 in existing facilities and laboratories that can already do bulk protein analysis well, quantitative bulk proteomics, I think all of these laboratories and facilities that can do quantitative bulk proteomics should be able to implement Scope 2. So there are no major additional bottlenecks. There is one disadvantage compared to single-cell RNA sequencing, which at the moment is at the level of throughput. 10x Genomics has made it possible to analyze order 10,000 single cells in a relatively convenient way in a single sample prep, and of course much larger number as part of higher scale projects. And this is still more challenging to do with Scope 2. The throughput of Scope 2 is much more comparable to the multi-well plate-based approaches such as CellSeq, SmartSeq, and so on. And to some extent, this reflects the current state of the field. It's not a limitation of mass spectrometry proteomics or single-cell analysis. It is simply the level of throughput that current technologies have achieved. And in fact, with the new multiplex data-independent acquisition framework that we've introduced, we believe that we can get to analyzing the proteomes of 5,000 single cells per day per single instrument, and potentially scale that even further. So the opportunity certainly exists to increase throughput substantially. Though at the moment, that is a relatively weak side when compared to the more mature droplet single-cell RNA sequencing methods.
Absolutely. And you've just mentioned analysis there. We're entering the data revolution, a huge amount of data, potentially overwhelm might be the word some people use. Machine learning is often touted as the solution. How realistic and attainable is it in your field? What are the limitations we might see?
Data analysis plays a very, very important role, and many of the gains that we have made, especially with the FlexDA approach, have critically depended on introducing new data analytics. And the same actually with Scope 2, where we took advantage of a Bayesian framework to incorporate additional features in determining peptide sequences, such as retention time. And data analytics certainly hold much promise to further interpret the data that we collect, of course, for downstream analysis as well, because simply quantifying protein abundances or interactions or modifications is just the beginning of the project. It's not the end goal. The end goal is biological interpretation, which again requires various types of data analysis. I tend to see data analysis as being a very exciting and productive component of what we do. I don't see it as being the limitation in any way. It is true that we generate gigabytes, sometimes terabytes of data, and those need to be analyzed. But fortunately, we have access to clusters that make this analysis quite doable. And I would say that data analysis in terms of the volume of the data is certainly not the bottleneck. Current algorithms are not extracting as much from the data as I believe we can extract. So one can say that they're limiting, and this is the glass being half empty. But the half full part of the glass is the opportunity to advance those algorithms so that we can interpret a lot more from the data. And I think that's a very exciting opportunity. So big data is certainly a data interpretation, data analysis, machine learning. They're certainly very important part of the field. And I think that we'll see many of the advances that ahead of us be driven or at least aided by improvements in data analysis. And we are certainly not limited by computational power at the moment.
Thank you so much. And the Slavov laboratory, you seek to coordinate among protein synthesis, metabolism, cell growth, and differentiation. Can you run us through some case studies of what's come out of your lab and the work you're producing?
So we identify ourselves with the systems biology community, where we try to understand what are some of the principles of biological behavior, emergent behaviors. Of course, we do many molecular studies, and we're interested in mechanistic exploration of what underpins biological functions. But oftentimes, we see that this is not a simplistic picture of a single protein or a single transcription factor doing all of the regulation resulting in a cell fate change. It's a more complex system that is underlying life. So I can share with you a couple of specific projects, recent projects that have given interesting results. One of these is we studied a process that is quite relevant to the beginning of my life and the beginning of your life, which is what first breaks a symmetry as a zygote gives rise to a 2-cell embryo and a 4-cell embryo. when do these cells become different? And for decades, developmental biologists believed that at the two-cell stage, the two blastomeres making up human mouse embryos are similar, perhaps identical, indistinguishable. And more recently, there has emerged evidence that they have different functional potential to develop into different fates, different bias. But it had been very difficult to identify molecules associated with these different developmental bias, despite the fact that multiple groups have applied state-of-the-art single-cell RNA sequencing to study these cells. There are very few RNA molecules that show bias between those two blastomeres. And when we approach the same question by measuring proteins instead of RNA molecules, we found many hundreds of proteins that were systematically and consistently differentially abundant between two-cell stage blastomeres. And we were able to find functionally coherent sets of proteins that follow that pattern. And furthermore, our data strongly suggested that what's breaking the symmetry in those early stages of our development is protein degradation and protein transport. And in retrospect, perhaps they should have been more clear, because in those early stages of development, transcription is not dominant, it's not very active. And therefore, the way that cells distinguish each other and break their symmetry is by working with the proteome that they have. They can either degrade or transport proteins to different location of the zygote, and then the two-cell stage, the blastomeres at the 2 and 4 cell stages. So that's a project that was quite encompassing in terms of the different biological experiments that we did, both functional and analytical, and clearly indicated a biological system or process where the most exciting and interesting processes happening would have been missed, and in fact had been missed when the investigation was focused just on measuring RNA molecules. Another recent project is studying polarization in primary macrophages in a model system that many people have traditionally considered as being highly homogeneous, and in particular, these are bone marrow-derived macrophages. Macrophages, while in general cells that are well known for being polarizable and wearing different hats, when in certain conditions, as when derived from bone marrow, at least people tend to think of them as not having a lot of single-cell heterogeneity. So we analyzed those cells, either unstimulated or LPS stimulated, and to the surprise of our collaborators from the Zanoni group at Harvard Medical School, we found that there are substantial molecular differences, even within a treatment group, even before these cells were treated with lipopolysaccharides. And of course, we were surprised, and especially the Zanoni group was very surprised by that, because they were convinced that we're not going to find any differences there. So then the question emerges, are these differences just some molecular fluctuations that have no biological significance, or do they have any relevance for the biological functions of these cells? So we explore that by further measuring endocytic activity, the ability of these macrophages to take up particles, fluorescently labeled dextran particles. And we found that the molecular differences that we found are strongly coupled to function differences. Indeed, there was 30-fold variability in endocytic activity of these primary macrophages. And another aspect that also highlighted the benefit now of using protein analysis as opposed to just RNA analysis in this system was our ability to measure proteolysis in this system. It is well known that many biological processes, including macrophage polarization, are regulated in part by regulated proteolysis. And by mass spectrometry, we can specifically detect the proteolytically cleaved products from that regulatory signaling, and be able to associate this with inflammatory or pro-inflammatory processes, inflammatory or anti-inflammatory. And of course, that's a level of regulation that is very difficult to measure if one is simply sequencing RNA molecules.
Thank you for sharing these fascinating case studies. And to link back to something you were talking about earlier, about reproducibility and accessibility, I know you also have a great YouTube channel with lots of your scientific presentations made available. I'd love to hear your thoughts on open access research and the importance of cross-disciplinary communication.
I think this is an example of a win-win strategy. I think that open research is very beneficial for the community, but also very beneficial for groups who practice it. Because in a number of ways. One way that benefited us very significantly in the early days is in establishing credibility of this emerging field, where we propose that we, the newcomers, can do something that the established leaders in the field couldn't do or claimed wasn't possible to do. And that, of course, resulted in a lot of skepticism. And part of what helped overcome this skepticism is the fact that colleagues from other laboratories downloaded our data, repeated our analysis, and obtained results that were qualitatively identical for all practical purposes to what we had done. And while reproducibility is not the same as accuracy, this ability to reproduce our results lended a very large degree of credibility to what we had done, and it was very, very healthy for the field. Another example of this benefiting us is, I think it sets the standard and the bar high for all of the students and postdocs in my group, so that if they make the work easy for others to reproduce, it also becomes very easy for them to introduce new data to their pipelines and easily revise their papers in the process of peer review. Which is unfortunately not the standard in the community. I was recently an editor for a paper where a reviewer suggested a very appropriate change to the data processing of the paper, and the authors responded that they couldn't do that because they would have to regenerate their figures, and that was very difficult for them to do and justified not doing it that way, which is quite pathetic, frankly. As a justification for not improving the analysis. So I think it takes more time to begin with to establish a reproducible pipeline of data analysis, but in the long term, it actually saves you time, because usually for impactful papers, one has to revise the figures multiple times. And if everything is set up in a way that allows to make these revisions to be done simply and quickly in the long term, it enables to benefit from a constructive feedback from reviewers, as in the case of the paper for which I served as an editor, and also allows to incorporate new data for others to build upon it. And ultimately, that's why we do science, in my view. That's perhaps a bit more idealistic, but I can do a lot of interesting things in life, and my time is very precious. Why spend my time on something that is not going to have impact, that other people cannot build upon and use it? It just doesn't make sense to me to even do that. It's not worth my time. If I'm going to spend my time in doing science and research, I might as well do it in such a way that it really drives the global research enterprise. It really provides useful resources for others in the community to build upon. So to me, it's obviously the right thing to do. And I think it's right not only philosophically in this sense, but I think practically it has helped us in a number of ways to attain high visibility and to help the field that we helped start.
Incredibly valuable, of course. And what do you think is the end game for these technologies and methods we've discussed today? What do you want to see in the future?
I would love to see the technology being so accessible and so inexpensive that every biological project that can benefit from doing single-cell protein measurements can do them as easily and perhaps more cheaply than currently people can employ 10x genomics for RNA analysis. But the other aspect for that is not only making it cheaper and easier, but extending the reach of what we can measure, because measuring proteins expands the scope of our analysis, but it's clearly insufficient. We want to be able to measure protein interactions, protein modifications, protein localization in the cell, protein activities, protein conformations, all of these very important layers of biological activities, biological regulation, that in principle, our technologies can be extended to make accessible. They currently cannot do all of these things. We have not yet done it, but I see no reason why this cannot be developed. And I think it's going to be a very exciting path of technology development towards achieving these goals. And once we have that technology, I think we'll be in a much better position to catalyze a more mechanistic approach to single-cell biology, not only to identify different clusters of cells and to describe differences in cell states in different pathophysiological conditions, but to be able to measure the molecular processes that ultimately underpin those different stages and contribute to either health or disease.
And to finish up, we're talking about being excited about the future. What are you excited about for the upcoming Triomics Summit?
I'm excited in networking with colleagues from different fields who can benefit from using the technologies that we have helped develop, implementing them to their problems, and joining the field. I think we need experts with different backgrounds. We need very interdisciplinary community to join and help realize the potential and the promise of single-cell proteomics. We cannot do this alone. We need a broad spectrum of expertise from different fields.
Thank you so much for joining me today. I've learned an incredible amount, and I know I want to hear more sneak peeks about what's coming out of your lab as well. But thank you for joining me today.
Thank you for inviting me. I enjoyed our discussion.
Hello everyone. If you enjoyed this video, then make sure you check out some of our other videos in our series by subscribing below or going to our website, frontlinegenomics.com. I hope you enjoy.